Have you ever really looked at a plant and just wondered? I mean, what if I told you that hidden inside a single leaf, or even a single tiny cell, is the complete blueprint to grow an entire new plant? Or heck, an entire forest? Well, today we're diving into the incredible science of plant tissue culture, a technique that lets us do exactly that. It's wild. So here's our game plan. We're going to start with the plant's secret superpower that makes this whole thing possible. Then, we'll flip through the lab manual to see how it's actually done. We'll even peek at the perfect growth recipe. And from there, we'll get into the really mind-blowing stuff, like creating virus-free crops and, you guessed it, growing life-saving medicine right there in a jar.
All right, first things first. What's the fundamental magic trick that lets us grow a whole plant in a lab? Well, it all boils down to this one-of-a-kind ability that most plant cells have locked away inside them. So let's just ask the big question straight up. Can you really grow an entire forest from just one leaf? I mean, it sounds like something straight out of science fiction, right? But the answer is very real, and it lies in a biological concept that really sets plants apart from the rest of us.
And the answer is a huge, resounding yes. It's all thanks to this one word, totipotency. Think of it as a biological superpower. It's the amazing ability of just one single plant cell to go, you know what, I'm going to become a whole new plant again, and then actually do it. It's the key to this entire process.
And, this idea isn't brand new. The whole concept was first figured out way back at the start of the 20th century. A German scientist, Gottlieb Haberland, was the one who first realized this incredible potential, and he basically laid the foundation for everything we're talking about today.
Okay, so we've got the what, totipotency. Now let's get into the how. It's time to open up the lab manual and see how scientists take this incredible theory and turn it into actual living, breathing plants.
The whole process is actually a pretty clear series of steps. First, you start with what's called an explant, just a tiny piece of tissue from a leaf or a stem. Next, and this is super important, you have to sterilize it to get rid of any pesky microbes. Then, you place it on this nutrient-packed gel, where it grows into something called a callus, which is basically a blob of blank slate cells. From that blob, you can coax it to grow shoots and roots, and before you know it, you have a tiny little plantlet that's ready to be introduced to the outside world.
And this is what it looks like in a real lab. Just look at all those little plants, all growing happily in their own sterile jars. It's kind of amazing to think that every single one of them started as a tiny little speck of tissue.
Now check this out, because this is what's really incredible. We're not talking about years of growth. Look at stage 1 on the left. That's the little piece of tissue you start with. And then, just a few weeks later, bam! Look at stage 2. You've got all these new shoots popping up. It's an unbelievably efficient way to make a lot of plants, fast.
But of course, those little plants can't just grow on hopes and dreams. They need food. They need the perfect environment. So, what exactly is in that gel they're growing on? Think of it like the ultimate 5-star, custom-made meal for a tiny plant.
That growth medium is basically a super complex cocktail. You've got all the essential building blocks, macro and micronutrients, plus vitamins, a sugar source for energy, and the really critical ingredient, plant growth regulators. Those are like little chemical messengers that tell the cells what to become.
Okay, so for all you science, botany, and pharmacy students out there, this is where it gets really specific. This is not just some random soup. Every single element in that recipe has a critical job. Nitrogen is there to build proteins. Calcium helps make strong cell walls. Iron is crucial for energy transfer. It's this perfectly balanced formula, and if you mess up just one ingredient, the whole process can grind to a halt. It's that precise.
So it turns out plant tissue culture isn't just one single method. It's more like a really sophisticated toolbox, There are all these different techniques, and each one is designed to do something very, very specific.
So let's crack open that toolbox for a second. You've got things like callus culture, which is great for growing huge masses of those undifferentiated cells. Then there's stuff like anther culture, which is a really clever shortcut for plant breeding. But the one we're going to zero in on is meristem culture, because it solves a massive problem in agriculture. Each one of these is a different key for unlocking a plant's potential. So let's talk about one hugely important job for this technology, something that affects food all over the world: making plants that are completely virus-free. A lot of our most important crops can get hit with viruses that just devastate harvests. And this is where meristem culture is just brilliant. See, the meristem is the super actively growing tip of the plant. And for some reason, it grows so fast that viruses can't keep up, so it stays clean even if the rest of the plant is sick. So scientists can just snip off that tiny,
clean tip, grow it in the lab, and regenerate an entirely new plant that is 100% healthy. It's like giving farmers a fresh start.
And that brings us to the final chapter, and maybe the most exciting one of all, especially for anyone interested in pharmacognosy. We're talking about using this technology to grow powerful, life-saving medicines.
Think about it. So many of our essential drugs come from compounds that plants make naturally. But that presents a huge challenge, right? What do you do when the plant is endangered? Or it takes decades to mature? Or it only grows on one specific mountain halfway across the world?
Well, this is where plant tissue culture becomes an absolute game changer. Take Taxol, a really important anti-cancer drug. It originally came from the bark of the slow-growing Pacific yew tree. But now, we can grow the cells that make it in a lab. The same goes for painkillers like morphine from poppy cells. We can create a sustainable, controllable, and reliable supply of some of our most vital medicines without ever having to harvest a single plant from the wild.
So, from a single cell to a whole plant, and from a simple test tube to a life-saving drug, plant tissue culture is just packed with potential. It really makes you stop and think, doesn't it? With millions of plant species out there, each with its own unique chemistry, what other cures are just sitting there, waiting for us to grow them in a lab?